So good afternoon, everybody. I'm delighted to welcome you all to the CF AMR Network launch event today. So for those of you who don't know, I'm Paula Summer. I'm Head of Research at the Cystic Fibrosis Trust. So the CF Trust is a UK-based medical research charity that funds CF research and advocates for people with cystic fibrosis. So we know that through our various involvement activities, which include a research priority setting partnership with the CF community, that infection is and remains a priority for the CF community. And so the Trust recognized this and seized the opportunity to partner with Medicines Discovery Catapult to form the CF AMR Syndicate. So the overall ethos of the syndicate is to help accelerate the development of new anti-infectives for cystic fibrosis. And today you're going to hear a little bit about what the syndicate is, its aims and objectives, and its activities, one of which is this network event. So you'll hear about that in a little bit more detail. You'll get some insights from people from industry, from academia, and people with cystic fibrosis who are already involved in syndicate activities. We're then going to have a small collaboration showcase where you'll hear from 4 presentations from people who are perhaps looking for possible collaborations. And then we're going to have some breakout sessions where we hope that you will help us shape our activities by telling us how we can help you. So without any further ado, I'd like to introduce Dr. Beverly Isherwood, who's the Program Manager for Syndicates at MDC. So over to you, Bev.
Fantastic. Thanks very much, Paula. So yeah, it's a pleasure to be able to share with you a little bit about the syndicate and our goals and what we've been working on. So if we could go to the next slide, as Paula says, we have there's a need within the CF community for us to develop better and more antimicrobial drugs. This isn't a unique challenge across many different diseases and conditions. There is a need for more antimicrobials, different types of antimicrobials that can really help to treat these often difficult to treat pathogens. And it is true that our research and development is really struggling to keep pace with the emergence of these pathogens. And there are now, and increasingly so, a number of different initiatives that are emerging to tackle this. With our initiative, the CF AMR Syndicate, what we're looking to do is to really help accelerate the translation of new CF antimicrobial treatments to the clinic through collaboration. And I'll just share with you on the next slide a little bit about where we've come from and how we go about doing that. So as Paula mentioned, we formed a partnership between the Medicines Discovery Catapult and the Cystic Fibrosis Trust. And the syndicate came into existence in late 2019. And we then have worked with a huge number of people and experts across the community in industry and academia, clinical collaborators, people with CF and their families, to really help identify the areas of patient need, and then to leverage that cross-sector expertise to catalyze new drug discovery efforts that can really help accelerate new antimicrobials in this space. And if we move to the next slide, what that's looked like is some of our progress to date since we've been working. 2 of the key challenges that we identified and have focused on in the first instance have been around the fact that although there's a lot of global activity in this space, there are still challenges around innovators looking to access clinical samples to test their promising antimicrobial compounds. There are also challenges with what is that preclinical screening pathway, what assays and endpoints are needed to really help progress these programs. And so off the back of that, as a whole community, we've worked together and we now are building a toolkit of capability, resources, expertise to help support CF antimicrobial discovery and development. So you can see a few examples on the slide here around a biorepository, guidance documents around key strains and isolates that are important for CF, a PIPE CF project, which you'll hear more about from Dr. Joe Fothergill in a few minutes, really around optimizing and mapping that preclinical screening pathway, similarly around being able to use artificial sputum media to help accelerate that preclinical screening as well, and then CF infection target product profiles. So let me just tell you in a bit more detail about 2 of those, just to bring them to life a little bit more. So on the next slide, this is one of our enabling resources. It's a newly launched project that is the UK CF infection biorepository. And here, what we want to do is to build and strengthen this access to these high quality, clinically relevant samples that will enable people from both academia and industry to test their new treatments. This is a collaboration across 8 legal entities alongside the Medicine Discovery Catapult and the CF Trust. And it's a 3-year program, which we hope will lead to a sustainable biorepository that is beneficial for the whole community. We will talk about this in one of the breakout sessions, so I look forward to joining some of you in that to talk in a bit more detail later. But that's one example of a new program launching now from the syndicate. The second slide that I've got here, a bit around enabling tools, is work we've done with many people around developing patient-focused target product profiles. Now, these, for people who aren't aware, is around providing guidance for drug developers on the key characteristics and requirements. And the key thing here is that we began this process with a focus group, talking and meeting with people with CF. And that was really to make sure that the needs and priorities of those people are addressed in the drug development pipeline, specifically thinking about things like the burden of care, burden of treatment. We've also then engaged internationally with clinical groups, clinical teams, industry, academia, and are in the process of refining. We held a virtual symposium earlier in the year to really help build additional expertise and insights into this target product profile, including regulatory input. And we're now in the process of finalizing these, and we hope to make them available to the community in the very near future to help guide development. So then, next slide, please. This is the core team, and just a chance to say a big thanks to everyone here, but also for all the people that we've engaged with to date. It's fantastic to be able to come together and start to develop these tools together collectively. And then I've just got one last slide talking a little bit more about the future, our current and future focus for the syndicate. So I've shared with you a little bit about our tools and resources. We want to continue to build those, and we'll be guided by the needs of people that come and tell us what's really needed. We also want to really identify and then support CF antimicrobial programs using these tools and new ones yet to be developed, and to really help facilitate and enable both of these things. This is the reason for launching the CF AMR network today, to really facilitate that collaboration and knowledge exchange. So that's a little bit about the syndicate. What I'll do now is to hand over to Jessica Lee from the Medicine Discovery Catapult, and she'll be able to tell you a bit more about the network and what we hope to get from it.
Great, thank you, Bev. Would you mind spotlighting me? Because I don't think I can spotlight myself. But thank you, everybody, for attending the meeting today. So, oh, there you go, you can see me now. Thank you so much for joining. I'm really pleased to tell you a bit more about the CF AMR network, which will now be launching to bring the CF AMR community together and to really start facilitating collaboration, knowledge exchange, and driving progress in this area. So next slide, please, Seema. So I thought I'd start off by talking a bit about the CF AMR landscape. And I think we're quite lucky because we straddle 2 different therapeutic areas. So we have cystic fibrosis, which is well served, especially by the charity sector, which really cares for the needs and priorities of people with CF, engages with clinicians, academics, and industry to try and drive progress in CF. And in particular, the Cystic Fibrosis Trust, who is our partner in the syndicate, has key assets and tools that are really useful for drug discovery in this area. So that includes the CF registry and the Clinical Trials Accelerator platform. And then in the antimicrobial resistance space, there are various networks and consortia, and you can see some listed here, as well as accelerators or funding opportunities, including organisations like CARBEX, who I'm sure we're all aware of, and newer opportunities like INCATE. And what we hope the CF AMR network will do is really bring together these 2 communities, so we can really ensure that those people who are developing antimicrobial therapies can access CF expertise, and they can ensure that their therapies are meeting the needs and priorities of people with CF, but also the other way around, so people who are in CF research can use their research to the maximum impact when it comes to developing new therapies. The next slide, please, Seema. So why do we want to create this network? Well, we want to tackle the big challenges in CF and antimicrobial resistance. We held a symposium, which Bev mentioned earlier this year, where we tried to understand the key challenges in this landscape. And they were things like developing a better understanding of pulmonary exacerbations, developing diagnostic tools, predictive biomarkers, drug delivery devices and reformulations, and also novel drug combinations. And what we're hoping that we can do is start to catalyse focused efforts in these areas through the network. Next slide, please, Emma. And we're not doing this in isolation. We're doing this together with the community. And there are key organisations that we'll be working in partnership with to deliver on our aims. And that includes the National Biofilm Innovation Centre, or NBIC, which is a community of biofilm researchers in academia and industry. They facilitate collaboration, they run funding calls, and they really kind of progress innovation in the field. There's the Gw4 Amr Alliance, which is a new alliance which brings together interdisciplinary researchers across Bath, Bristol, Cardiff and Exeter to tackle AMR. And within this, there are 3 CF centres with a population of over 1,000 people with CF. So it's a great source of collaboration and opportunity. And also there's INCAPE, which I've already mentioned, and this is a new incubator for antibacterial therapies in Europe, really supporting the startup AMR community, which obviously includes the CF cohort as well. And we actually have people from each of these organisations on the line today, so feel free to type your questions in the chat to ask a question, or you can see email addresses listed here as well if you want to get in touch directly. We are also launching a new funding call, thanks Eva, in partnership with WeShare Ventures, which will be coming soon. And this is to fund early stage CF, AMR, medicine discovery startups across Europe. So if you don't already know about that, feel free to get in touch with me and I can tell you more information after the event. Next slide, please. So I talk about trying to bring together the CF AMR community and drive innovation. But what I want to highlight is that the CF AMR community is already innovating. So we're exploring phage therapies, we're exploring therapeutic bacteria approaches. You'll hear from the talk soon. We've identified new antibiotic classes that could be suitable for CF and antivirulence approaches. And actually, we believe that the CF AMR community can lead the way in antibiotic and antimicrobial development. And what we want to do through this network is to help you. Next slide, please, Emma. So the network, all about catalysing innovation and working in partnership to bring together a critical mass of expertise and capability in the area. Through that critical mass, we want to drive and facilitate introductions between network members. We want to ensure that any new entrants into this area can accelerate that integration quickly. We also want to, working closely with the Cystic Fibrosis Trust, unlock opportunities to involve people with CF. And we'll hear from Lorna soon on how important and critical that is. And we also want to enable knowledge exchange and networking. So we'll do that in a few ways. One is through our website, which we'll be launching soon, and our newsletter, where we'll host blogs and news and resources. So you can come to us with news that you want to share, or you can also come to the website looking to read about some news. We'll be running events like this, workshops and focus groups with the aim to promote knowledge exchange, but also problem solving in some potential new areas that maybe required a concerted collaborative effort. And we'll also, as I mentioned, be running that networking and signposting function to really catalyse collaboration and open up this community really. So what I wanted to do now is move on to host a short panel discussion. And our mantra of the day today is short and snappy. So we're inviting up Lorna Allen, Jo Fothergill and Deb O'Neill to tell us a bit about themselves and how important collaboration is for them in antimicrobial development. We'll then also welcome back to Bev I always get mixed up between Dev and Bev. Well, welcome back, Bev. And we'll do a bit of a Q&A session. So if we can stop sharing the slides now, Seema, I think we can just spotlight. Yeah. So maybe Joe, you've unmuted first, so I'll come to you first. If you could just introduce yourself and why collaboration is important for you in this area.
Hi everyone, I'm Jo Fothergill from the University of Liverpool, and I've been part of the syndicate for just over a year now. And I think one way that I can highlight that collaboration has been really important in this area is that about 18 months ago, a group of us, many of whom are on this call, started discussions about a lack of a clear pathway to develop therapeutics in this area. And certainly the interactions with the syndicate in terms of identifying barriers and also putting in links with industry was really important in this application. And we were successful in getting an SRC, a Strategic Research Centre, funded by the CF Trust and the CFF, and that was announced just yesterday. And I think that makes it abundantly clear how important collaboration is in this area. Ultimately, I think we all want the same thing in this area, and that's to develop more therapeutics that are, are better and give more options for people with cystic fibrosis.
Thank you so much, Jo. Maybe Lorna, we can hear from you next.
Yeah, hi, I'm Lorna. I'm the involvement manager at the Cystic Fibrosis Trust. And as Jess mentioned earlier, I'm also a CF mum. My daughter's 23. And I was really thrilled to be asked to really facilitate some of the involvement activity in the development of the TPPs that Jess mentioned earlier. And we did that through the community involvement work stream that we've developed at the Trust. And this is a real opportunity for research teams and sponsors to tap into the knowledge that people with CF have of living with the condition day in, day out. And the insights that really came through in those early focus groups with the development of the TPPs were so striking. So it's the knowledge that goes beyond the clinic. So even some of the clinicians that were involved in those focus groups were hearing information and learning from people living with CF about aspects of the development of new treatments that they hadn't even considered before. So we all know that the burden of treatment is huge with CF. And when you're looking at developing a new treatment, the consideration around how that's going to fit into somebody's treatment regime alongside everything else they do. So it's not in isolation. What are their tolerances with regards to clinical trial endpoints, to the schedule of the clinical trial, and how that potential new medication could move into the care setting as well. So we're really pleased to be able to facilitate those conversations. We have an involvement group that's actually nearly almost 100 members now. We do one-off engagements, ad hoc engagements, and they're also there to review documents, say to look at trial design, particularly around the schedules, which hopefully will make recruitment easier, quicker, make retention in trials more reliable, and ultimately saves everybody time and cost. And as Bev alluded to, has the opportunity to accelerate development of treatments. I was just going to mention one of the other really strong successes that we've had with involvement, and say this really demonstrates the benefit of involving as early as possible. And this is with a study called CF Storm that has recently opened. And even before it was named, before it was designed, we were having focus groups with our involvement group members to find out what it really was that they wanted when we were considering reducing the burden of treatment. And we were really able to drill down into what it was that gave them the most difficulty in sticking to their regime, but what they were potentially prepared to sacrifice in losing some of that burden of treatment of their routine as well. And we're really pleased that that study has now been funded, it's been opened, recruitment has started, and we still have people involved in that and the TPPs, and now also the biorepository that Bev mentioned on the steering committees, say a continual collaboration with people with CF.
Thank you, Lorna. And I think the really important thing for us through the syndicate is to highlight the critical importance of actually involving people with CF in research. So thanks. And last but not least, our amazing chair of our steering committee, Deb O'Neill.
Just unmuting myself as well, Jess. Thank you, and hi everyone. Absolute pleasure to be here at the launch. It's a shame it's still virtual, but there we go. So I guess speaking from the industry perspective, my day job is the chief exec and founder of a biotech company developing a number of antimicrobial therapies, some of which will be for cystic fibrosis, so non-antibiotic antimicrobials and inhaled antifungals. You cannot develop a new therapy in isolation as a company, whether you're big pharma, whether you're small biotech. You know, it might take a village to raise a child, takes a syndicate and a very big network to develop a therapy, particularly a truly novel therapy. You know, huge paradox, which is crazy, the underinvestment in antimicrobials research, full stop, across the board. Shrink that down to the area that we're focused on in cystic fibrosis as well, you have an even bigger problem. So for any company, and particularly some of the exciting new companies that approach Jess, the other team members in the syndicate looking to get into this space, where do you go? You know, there isn't really a precedent. There's no roadmap. Joe mentioned the preclinical pathways, but it's very difficult for anybody in this area to know how to do it, where to go, what to do. So collaboration is absolutely key. You know, being part of the syndicate has benefited Novabiotics, and it's all really about access to, it's actually 5 strands. We have a really novel form of nucleic acid. We've gone beyond a triple helix. So it's not just as part of the network, the other companies that are involved, as you've seen from the earlier slides, the fantastic academics that are involved, the clinicians, the patients, and I think now, as you've heard, potentially some road mapping to funding for companies as well. So the ability to have that access to knowledge sharing, resource, and the tools is absolutely critical. So hopefully for any company to be able to accelerate their journey into developing novel therapies, it allows them to get into the ecosystem that they need, have all the connections that are absolutely vital. And I really hope that the outcomes that we see from this are in the future some huge leaps forward, potentially into some new therapies, much needed antimicrobials in CF.
Thanks, Deb. It's always good to hear that what we're doing is important. So thank you. Great. So maybe what we can do is first, I'd be really interested, Joe, if you could tell us a bit more about the SRC that you just mentioned that was launched yesterday.
Okay, so this is a project that we've called Pipe CF, and it really has been born out of identifying some of the barriers. And I think, as Deb has highlighted, that you know it's difficult for an individual or an individual company to go through this process of developing and optimizing appropriate models, and it's difficult to know which models are appropriate for the development of these types of therapeutics. And so I think you know it's really great that this is an academic-led consortium, but with all of the interaction with industrial partners and regulators. And so the idea behind it is that the barriers that we have identified in collaboration with you guys are about using relevant strains, certain influences like polymicrobial influences and biofilms, the importance of potentially looking at host responses depending on the therapeutic, and the sort of robust and validation of in vivo models, which can sometimes be a bit problematic in that area, and certainly a lot of discussion around that. And of course, there's the changing landscape of both treatment of people with CF, but also the changing landscape of the types of therapeutics that are coming through. And all of that together means that there isn't really a clear pathway there's lots and lots of options, and I think sometimes that can be a barrier to the development. And what we really want to do is develop a clear pathway that is an open platform, accessible to academia and to industry, in order to hopefully accelerate more therapeutics through this pipeline. And ultimately we'd like to increase the success. What we want to know is, will that therapeutic work in this group? And that's a really challenging thing to address, but we're hoping that we can get closer to that point.
Thanks, Jo. Please do feel free to raise your hands as well if you've got a question. I do have one here in the chat. So how early on in drug development would you involve people with CF? This is probably for Lorna. And how would you go about doing that?
I'd say from the point of concept, really almost as soon as you're having the idea and the thoughts, and maybe even if you don't necessarily know how to progress that or how you're going to progress that at that stage, I'd say that's the ideal opportunity to involve people. And you can do that by contacting myself and the involvement team at the Trust. As I say, we are there to work as that conduit between yourselves and our community members.
Thanks, Lorna. And Deb, you've involved people with CF directly through your Linavax program as well. And how important has that been for you?
Yeah, absolutely. So I think, again, as I said, coming from the industry side, you can't develop new therapies in a bubble. If you do, you'll probably end up with some very difficult conversations with the regulators and what you think is a therapy that might be fit for purpose. So, I think, as I've said to you, Jess, it would have been fantastic if the syndicate had been around for us a few years ago as well. So just the ability to tap into everything that we can hopefully provide for new companies coming up with novel therapies like Linavax. As Jo touched on, it's probably going to be multiple pathways. No two compounds are going to be the same. What we're developing is very different from probably the next company that might approach the syndicate for some advice. So I think just that resourcing and arming companies with the best information from the best group of people, and as I've said, covering the best academics working in the field on various aspects of CF infection, the clinicians who are looking after the patients themselves. So I think, having the Trust on board as partners is essential. So all of that's vital. And it still will be, where we're quite advanced now, going into registration studies with Linavax. Our antifungal is at an earlier stage, but we'll still tap into the expertise that the syndicate provides. So yeah, it's very important.
Yeah, and I guess that's a two-way street as well. So I think you're in obviously the fortunate position that you've got all this expertise in-house, and you've been through the pathway. So how can companies, newer companies, actually learn from companies like Gw4 Biotics who do have a lot of expertise? Definitely.
So I think that the more companies we can get on board to share their expertise as well and their journey. So for example, we've been further along down the regulatory path, so we've had those discussions with the regulators and various regulators across the world. Some of the other companies have had discussions with other regulators that we haven't had, and we were able to share that experience with other companies coming through, which is critical. And hopefully keep that information fresh. Signposts, I know the piece on TPPs at the moment, even experts and consultants that the companies have worked with along their journey, particularly smaller companies, if we can bring them back in and help all the companies that are tapping into the network as well. So that is critical, because as I said, it is a very small world. So I think the more we can rely on each other's experience to help smooth the path for other companies, definitely the better.
Thanks, Deb. We've got a note in the chat from Stephen, which is great, saying that Lorna helped him to involve people with CF in their strategy discussions, and it was very inspirational. So that's really great to hear. I think maybe one final question from me, which is for Bev. I know that you're running a session on the CF-AMR biorepository later, but for those who aren't attending the session, how would people go about getting in touch with the CF-AMR biorepository, and when can they? Is it open now?
Yeah, so it is. We're open for business now. The way to contact us would be just through email, either directly to myself or through the CF-AMR email address, which you've seen on the slides. You'll see in a following email that comes out after our network event. And then what we'll do is to really help understand your requirements or your interests. It may be that you have samples that you'd like to be part of the network, or you'd like to know more about it, and then we can connect the right people together, hopefully. So yeah, we're really excited. It's a very new initiative for us, and we're really hoping that we can get the samples, data, the expertise, and match make those with the people that need them. Yeah.
Great, thank you. So if there's no other questions, I think we'll move on to our collaboration showcase, which Andres, I believe you are chairing for us. So we'll try and spotlight you, Andres. Thank you so much. Deb, Lorna, Joe, Bev.
Hi everyone. Thanks Jess, and obviously all of the Medicines Catapult, Discovery Catapult for organising the meeting. We've got 40 minutes in total for this session, where we're hoping to showcase some really cool abstracts that we had. People have submitted really high standard, and we're delighted to have 4 of the top speakers from the top 4 abstracts to give talks. The aim are to give very short and snappy 5-minute overviews of the research. I've had a warning from Jess that people who run over the 5 minutes will get interrupted with a emoji smiley face. I don't know what that means. So without further ado, what I would say is please post your questions on the chat or raise your hand, and at the end of each talk there'll be a couple of minutes of Q&A. So without further ado, it's my great pleasure to introduce Professor David Greenberg from UT Southwestern, who is currently developing novel lead compounds to treat multi-drug resistant Pseudomonas infections with a focus of the CF population. David, over to you.
Okay, thank you so much. Thank you to the organizers for allowing me to speak. I'm thrilled to be here. Just as a way of background, I'm an infectious diseases physician scientist. On the clinical side, I do transplant infectious diseases. We have a very robust lung program, and many of those lung transplants are CF related. And my lab focuses on antibiotic resistance, either on the therapeutic end or the diagnostic end. And we've been working on therapeutics for a number of years. And so I'm going to distill a number of years' work down to 5 minutes to keep it snappy. So next slide. So my lab focuses on developing novel antibiotics. We're really trying to think about new therapeutics from a completely different standpoint. And I don't have time today to talk about all the reasons why we're thinking about pathogen-specific antibiotics, but as everyone here knows, and I'm preaching to the choir, part of this is driven by trying to help circumvent some of the problems we have with antimicrobial resistance. So my lab makes pathogen-specific antibodies using a large molecule approach, specifically with Antisense. Next slide. So for those who don't know, what are antisense molecules? Very briefly, these are short oligos, nucleotides, either DNA or RNA mimics. The ones that we utilize are designed to bind to complementary RNA. We are specifically using a chemistry called phosphorodiamidate morpholino oligomers, a very long word. We'll just call those PMOs for now. And these are normal nucleotides, A's, C's, G's, and T's, but they have a morpholine ring as a backbone, which is critically important. These are uncharged molecules compared to other antisense chemistries. They are resistant to DNases and RNases and highly stable. And in fact, people have been using antisense and different antisense chemistries for many years across a variety of disease states, including early work in thinking about these as antibacterials with very poor success. And it turns out that these small naked PMOs have a very difficult time penetrating bacterial membranes. So some of the innovations that have occurred over the past 10 years or so with work from my lab and my co-inventor on many of these, Bruce Geller, who did a lot of the early work using antisense in E. coli, was the linking of these antisense molecules, sort of shown here graphically on the right, to various peptides. Now, these are synthetic peptides. They maintain many of the properties that normal antimicrobial peptides have in our body, such as in our saliva. When you link these to these oligos, the peptide is really the delivery vehicle for getting them into and across the cell wall and membrane. And so these are not antimicrobial peptides as a therapeutic. In fact, the ones that we utilize by themselves aren't really antimicrobial. It's purely a delivery vehicle. So by linking these peptides to the antisense molecule, we can target the mRNA of any bacteria that we're interested in. And I won't go through our whole program today, but we now have lead compounds across gram-negative space. So if you have a genome, you can target these to whatever gene of interest you want. And we have a number of programs, and we've published extensively on using these to target bacteria to kill the organism. So we're going to do what small molecule antibiotics do, just in a gene-specific, pathogen-specific way. We could use these to modulate antibiotic resistance. So that would be using what I'm now going to call PPMOs, so peptide conjugated PMOs, as an adjuvant to rescue traditional antibiotics. And I'm not going to talk about it, but you could also use these to modulate virulence. Next slide. So we have a robust program in Pseudomonas. This is probably our lead program, obviously a major pathogen in both CF and non-CF patients. I continue to see pan-resistant and multi-drug resistant isolates from our CF patients when I'm on service. And so over a number of years, we developed gene target leads. These are targeting either known or putative essential genes that have been discovered across gram-negative space. So for example, on the bottom is sort of a heat map view that's showing you MIC data across a panel of Pseudomonas isolates. And I'll just point out at the top are 3 gene leads. One is probably familiar to many in the group, LPXC. So that's an example of a lipopolysaccharide gene that other companies have been looking at for a number of years as potential antimicrobial targets. RPSJ is a ribosomal target, and then ACPP is an acyl carrier protein. What's interesting about ACPP is that is currently an undruggable target. So although it's an essential gene in Pseudomonas and it's an essential gene in a number of other gram-negative cells, there are currently no approved small molecule antibiotics against this particular gene. However, with this platform technology, it ends up being one of our robust targets. And in fact, I'm showing you 3 different gene targets with various peptides that are either attached on the 5' or 3' end, and we can develop MIC 90s in the low micromolar range. So ACPP, for example, is one of our main leads, and we have IC75s of 0.5 micromolar. Importantly, and critical for this program, it doesn't matter what the phenotypic background is of the Pseudomonas isolate, meaning the way that these get in and the way that they act are devoid of the background resistance rates of a particular strain. So I'm showing here a number of clinically obtained whole genome sequence strains, and it doesn't matter whether you're a lab strain like PA01 or a bloodstream isolate from a cancer patient that has metallo beta-lactamases like NDM1 or truncations in porins or VIMs or a variety of beta-lactamases, that you achieve similar MICs despite the phenotype. I wanted to point that out because we're interested in making antibiotics for multi-drug resistant pathogens. So that is a critically important part of the program. The other thing I'd like to point out is that these are active in biofilms. So in the top right, these are biofilms grown in an MBEC assay. The top right panel is a Pseudomonas biofilm that's fluorescing green, and the red is staining of the EPS, the biofilm, where we incubate the biofilm with a scrambled antisense molecule. So a made-up sequence linked to the same exact peptide, and you can see from above or from the side quite a thick biofilm. However, our ACPP, PPMOs are quite active, and this is biofilm reduction. So we grow biofilm for 24 hours, and then we dose these compounds Q8 hours, and you can see a dose-dependent response based on crystal violet staining on the left. Next slide. These are also active in vivo, and despite being large molecule compounds, they behave very much like traditional drugs. They show a dose-dependent effect in mouse models of pneumonia, which is shown in the top right, where we dose mice intranasally in an acute pneumonia model. And you can see at, for example, 15 milligrams per kilogram of our RPSJ PPMO, at 24 hours, it outperforms tobramycin at an equivalent dose. The bottom left panel is showing that these are active in vivo, even in XDR strains. So this was a contemporary clinical isolate that was resistant both to tobramycin and meropenem, where we dose in an equivalent way in this acute aggressive pneumonia model. So mice are infected intratracheally, and then we deliver PPMO either intranasally or systemically. And you can see here that the ACP PPMO was superior to meropenem or tobramycin. And then importantly for this group, I just wanted to highlight that we've actually been nebulizing these and aerosolizing these in the mouse version of a human nebulizer, where we infect mice again with an acute pneumonia model, and then we come back and deliver drug. And in fact, these are aerosolizable and reduce lung burden, at least in this model. Next slide. So I did not have a lot of time to talk, which is fine. I like being snappy, but I'll just point out that for us, this is not really just an academic exercise. This program now has 3 leads in Pseudomonas space. We have leads in Burkholderia. We are very interested in getting this to patients. And so we're in the early stages of trying to get this out of the lab. And I would be thrilled to talk with any of you who are interested in helping push this program forward. My email is right here, and I'm happy to take any questions. Thank you so much.
David, thank you. That was totally awesome. Can I remind everyone just to post questions on the chat or raise their hands, although I might have difficulty seeing those. So post the questions on the chat. David, can I maybe kick off? Can you say a little bit more about the mechanics of how the peptide nucleic acid conjugate gets through? I'm just having difficulty with the geometry here. Do you think?
So great question. You know, when I was younger and a medical student, I was always taught that antimicrobial peptides function by sort of non-specific disruption of the outer gram-negative membrane. That's probably partly true, but I don't think that is the complete story. And in fact, there probably is a level of active transport of certain peptide moieties across the cell membrane. Why do I say this? It turns out that not every peptide that works equally well in Pseudomonas, for example, would work to the same level in Burkholderia. And in fact, the peptides that we use in Burkholderia don't work very well in Pseudomonas. The other clue to this is resistance studies have been done, at least in E. coli, where mutants were made to PPMOs. So I mentioned that although PPMOs are active in XDR, MDR strains, like any other drug, that doesn't mean you won't develop resistance to that particular drug. You'll develop resistance to everything. It's just the mechanisms by which that happen are probably going to be different. And in E. coli, if you generate random mutants to E. coli PPMOs, and this has been published by Bruce Geller, you actually get mutations or find mutants in a particular peptide transporter. Okay. But what's interesting about that publication is if you take the same PMO and you link it to a different peptide, you can restore activity in the mutant. And that's critically important. And that's one way that we're thinking about our development program is how do you circumvent resistance? So now you could imagine a couple scenarios where the ultimate drug or formulation could be the same gene target with 2 different peptides, for example.
And is it known how many, David, is it known how many peptide transporters there are, what the repertoire is in gram negatives? Can you?
I think it's genus specific. So we have not found specific peptide transporters for each PPMO that is our lead. There probably is also non-specific uptake. So I think it's a little complex. Okay. It's going to be a combination of both.
David, sorry, I've just been moved on by Jess. Apologies to Miguel and Leshan for the questions. I'm going to ask Eshwa's question because it was something I was thinking about as well. So do the peptide conjugates to other molecules, do they conjugate easily to other molecules? And does it mean that you can get other stuff into bacteria?
The short answer is presumably yes. These are not complex conjugation chemistries. These are linear peptides. There's a whole family of cyclic peptides that can be more difficult to link to things. But yes, in theory, you could attach this to a variety of things.
Terrific. David, thanks again. There are lots of questions in the chat. I was wondering if you wouldn't mind, while we're listening to Freya's talk, just answering those individually. Thank you for everyone for posting the questions. David, thanks again for that. That's awesome. Moving on then to next, Dr. Freya Harrison. Is that all right? University, excellent. University of Warwick. So Freya's going to tell us a bit about ex vivo lung models, I think, for high throughput testing of antibacterial and antibiofilm agents. I think, Freya, you're ready to go.
Yep, that's great. Thank you very much, Andras. I wanted to give a quick introduction / update to the ex vivo model of CF bronchiolar biofilm that my lab's been working on for the last few years, really with the motivation of highlighting ways in which you can collaborate with us if you'd like to use this model for R&D. So could I have the first slide, please, Seema? Fab, thanks. So what we do is we take pieces of bronchiolar tissue from pigs that have been slaughtered for meat. So pigs are a great model for human tissue, and also by sourcing an animal that's used for human consumption, we essentially use a waste product from the meat industry, so our model is very 3Rs compliant. We cut out small 5 to 10 millimeter sections of bronchiolar tissue, and we combine this with synthetic CF mucus, sit the pieces in a 24 or 48 well microtiter plate, and we can grow really nice mature biofilms of key CF pathogens. So here I've just highlighted a really beautiful gloopy sleeve of Pseudomonas aeruginosa biofilm that's been grown for several days in this model. Once we've got mature biofilms of our bugs in this model, we can expose them to antibiotics or to candidate drugs or to different potential treatments or different environments. We can then use a bead beater to recover cells from the biofilm to enumerate how many bacteria we've got, or perform other downstream assays like assay the supernatant around the cells for secreted products like virulence factors. And we can also do some nice in situ imaging of the biofilm with light microscopy, with confocal microscopy, and also with EM. Could you click for me, please, Seema? Thank you. Just to show two examples of what we get in this model, you can see at the top a beautiful mature structured biofilm of P. aeruginosa around the lung tissue, where we can see this nice structure of individual cells embedded in matrix with channels in the biofilm. The way we find Staph aureus growing is particularly interesting. We find a little bit of biofilm-like aggregate on the tissue surface, but primarily in this model, the staph grows as biofilm-like aggregates of cells in the synthetic mucus around the tissue, which is actually quite similar to what you'll see in a biopsy from a person with CF. And it's very different from what you'd see in a mouse model where you tend to get invasive disease, which isn't quite so representative of the human situation. Next slide, please. I've just highlighted some published work, which is all open access if you'd like to look at what we've done to look at biofilm growth, virulence, antibiotic tolerance of some key pathogen species. We've got an open access protocol on the Journal of Visualized Experiments, where you can see a written protocol and a video demonstration of how you can prepare this model and use it in your own lab for high throughput antibiotic sensitivity testing of biofilms. And I'd just like to highlight two recent explorations we've had of P. aeruginosa biology in the lung model. Could you click twice for me, please, Seema? Sorry, part of the slides aren't showing. Fab. So most recently, one of my PhD students, Neve, has just completed a beautiful transcriptomic study looking at what genes Pseudomonas is switching on and off in the lung model and how that differs from if you just grow it in the synthetic CF mucus in vitro. And she's seeing some really interesting phenotypes and transcriptomes of the Pseudomonas in this model. We've also just this year published a study where we show really low penetration of antibiotics like colistin into the biofilm matrix when you grow Pseudomonas in this model. So just highlighting one CF isolate where we're getting it surviving a 10-fold higher concentration of colistin than you would measure as the MBEC if you used SCFM in a Calgary device. And we've shown that's because you get much poorer penetration of the antibiotic through the biofilm matrix when it's grown in the lung model. Next slide, please. So there's various ways you can interact with this if you want to use this model for fundamental biology work or for preclinical testing or later R&D. Thanks, Seema. So very happy to collaborate if you want to use our expertise and work together. So an example of that recently, one of Martin Welch's students made some beautiful multi-species biofilms using our model. We're also part of Joe's PIPE CF SRC, which will be very exciting. Very happy to go for joint funding with people. So I have a shared PhD student at Warwick with Seb Perrier, who's been making antimicrobial polymers and testing them in the lung model. And I'm part of a consortium in the UK and South Africa trying to optimize different antimicrobial peptides and see how well they work in high validity infection models. For more translational work, just highlighting some things we've done. Happy to train people in the use of the model. So I did this with one of Rowena Jenkins' postdocs, and he was able to get good ex vivo data on the ability of Manuka honey to kill Pseudomonas biofilm and synergize with antibiotics. And that data was very helpful to them in then moving into a clinical trial, which is currently running in Wales. Again, happy to apply for joint funding with companies. We've done that just very recently with Brenta. We've got some money to work with them to test some of their mesoporous materials for biofilm drug delivery. So I actually put in the grant application through funding at Warwick so that we could work together. And just finally, if you could do one last click for me, please, Seema. Thanks. If you would like to use this model on a client basis, we've worked with Perfectus Biomed to train their team so that they now have an ISO UCAS accredited version of our pig lung model in their lab for commercial biocide testing, and that's already been used by a client with a candidate CF drug for nasal delivery. And we can also work on a client basis in-house at Warwick if you prefer that to an academic collaboration. So that's a very whistle-stop tour. Thank you very much for your time, and I'm very, very happy to talk to people about ways you might like to work with us.
Freya, that's brilliant. Thank you very much. Brilliant timekeeping and really exciting opportunities for the model. I just wondered if I could ask something on the host side then. So what kind of, how faithful a model is the pig epithelium for the human epithelium? I presume quite, if the CF pig is so successful as a model. So
yeah, I would say the consensus is much better than a mouse. There's been some quite nice NMR metabolite profiling of different mammal airway surface epithelia and airway surface liquid, and the pig comes out as quite a good match for the human. But on the host side, one thing I would flag is that we have some residual innate immune response in the tissue from this model, but we don't have an adaptive immune response. So if people are specifically wanting to look at interactions with the host immune system,
right. But presumably you could stick macrophages on, can you? Pig macrophages? Yeah,
there's no reason why you can't add cell lines to this, or even add human cell lines to this.
Terrific. Question coming through then from Leshan. Freya, interesting work. Have you tried to use purified human respiratory mucus from the patients in the model?
We haven't actually. Thanks, Leshan, for your question. One of the things we wanted to do this year was to take whole sputum samples, whole CF samples, and use them in the model. Unfortunately, COVID has set that back quite considerably. But yeah, it would be really nice to take some lyophilized purified sputum and use that. I think the reason we've stuck with the Whiteley Labs implementation of the synthetic mucus is just because there's been some nice chemical characterization of it, and they've shown some good mapping of essential genomes and transcriptomes of key pathogens to what you see in sputum. So we've stuck with that as good enough for making it high throughput and affordable and achievable.
Thanks, Freya. And just to clarify, what generation of Airway are you using? Is that specified in the model, or?
So which part of the airway?
Yes.
So we dissect out. We can take either the main bronchi, or we can go quite low down and get the sort of like 2 to 4 mil bronchioles. It actually depends on which landrace of pig your butcher is slaughtering. Some of them have more accessible small bronchioles than others.
Do you see differences in the model? Presumably you do, depending on the airway caliber and characteristics, or?
That's a really interesting question. We've not specifically compared different parts of the bronchial tree from the same pig. We've shown good repeatability between individual animals when we take the same kind of bronchiole, but that would be really interesting to look at, sort of transect.
Wonderful. Freya, any more questions from anyone? Okie dokie, great, Freya, thank you very much. Moving on then. So next up is Dr. Kirsty Smitten from Metallobio Limited. Kirsty's a Royal Academy of Engineering Enterprise Fellow with a PhD from the University of Sheffield, and Metallobio is a University of Sheffield spin-out company developing novel antimicrobial compounds. So Kirsty, over to you.
So I'll just apologise in advance. My internet's a bit sketchy, so I've turned my camera off in hope that it won't fall out. Sorry in advance. Can I have the first slide, please? So antimicrobial resistance is globally responsible for 700,000 annual fatalities, and by 2050, this could result in 10 million deaths a year, costing the global economy 66 trillion. At Metallobio, what we're doing is focusing on drug development, but we're focusing on introducing a new antimicrobial class to the clinic. And we've done this through a radical new chemistry approach. Can I have the next slide, please? So the way we synthesize our compounds is through a modular synthesis. I usually describe it like Lego. So we have the ruthenium and then the central ligand, and they're the building blocks. And then we can add the outer building blocks, and we can change these to alter the properties of the complexes. And it's really easy to alter these, so we can make a whole library of these compounds. At the minute, we have a mononuclear series with the 1 ruthenium center and a dinuclear series with the 2 ruthenium centers. So we've isolated 2 leads that we're taking into preclinical development, and currently these leads are more active than clinically available antibiotics and include this on CF relevant pathogens. They retain this high activity on gram-negative, WHO priority and CDC urgent pathogens, so those that exhibit resistance to carbapenem, cephalosporin, vancomycin. They have multiple mechanisms of action, including a novel target, and they have little to no emergence of resistance in comparison to clinical antibiotics. So we've done evolution of resistance with 2 months of passaging on E. coli, A. bumannii, and Pseudomonas, and we see a 4 times increase in MIC for the dinuclear and a 2 times increase for the mononuclear. But when we compare this to other antibiotics, there's a 666 times increase in MIC for amoxicillin. So obviously we can confirm that there's no statistical difference there. There's a statistical difference there. So we know that we've got little to no emergence of resistance. We've also shown that the compounds have a high efficacy in vivo. So we've done an invertebrate model using Galeria melanella wax moth larvae. And the reason that we've used Galeria is they have an innate immune system that is similar to that you see with the human immune system. So there's hemocyte cells that can phagocytose bacteria, and they act like neutrophils and macrophages within mammals. We've tested this for A. bumannii and Pseudomonas aeruginosa. So we used PA2017 and then AB184. So AB184 is a prevalent clonal group in the UK for Acinetobacter, and the Pseudomonas strain was a clinical isolate that we got from the University of Surrey. And we saw that both compounds completely cleared the infection. And we monitored this not only through isolation of the hemolymph, but also through optical microscopy, which I'll show you later on. The compounds were found to be non-toxic up to 80 mg per kilogram in the larvae. And now we've more recently done an in vivo model and found that the compounds were non-toxic up to 30 mg per kilogram in a murine model. That was just the highest dose that we went up to within that model. We've also shown that the compounds have a large therapeutic window, both in vitro and in vivo, and they have broad spectrum capability. So obviously a problem with patients with cystic fibrosis is they get infections, and the infections are caused by a range of bacterial species rather than just a singular species. So it's good that the compounds have that broad spectrum capability. The compounds also, due to their modular synthesis, can be incorporated into different polymers, meaning not only could we use them as a systemic antimicrobial, but also as an additive within coatings. So we've tested them in a load of different polymers now, and they prevent bacterial biofilm formation at loading levels as little as 1%. Can I have the next slide, please? We've tested the compounds on around 150 different bacterial strains now, but I wanted to highlight a number of key strains. These are World Health Organization priority pathogens. Burkholderia and Pseudomonas are obviously relevant to cystic fibrosis patients. And for the dinuclear lead, we see activities of 0.5 to 5 micromolar, which is comparable to what we see within the wild type strains. The compounds also are active on bacterial biofilms, so they eradicate biofilms at concentrations similar to we see in planktonic cells, and then they also prevent biofilm formation. So we've done, again, a porcine model, but we haven't done a lung model, so we've done a porcine cornea model, and what we did is grew a Pseudomonas biofilm onto the porcine cornea, and then treated at 2 MBEC concentration of the compound, which was around 4.8 micromolar for the Pseudomonas strain. And we saw complete clearance of the infection at 2 times MBEC concentration for both the mononuclear and the dinuclear compound. The other good thing about these compounds is they're luminescent, so we can directly track where they are within the cell. So with the dinuclear, it has a multimodal mechanism of action whereby it initially targets lipopolysaccharides on the outer membrane and then targets cardiolipin on the inner membrane through electrostatic binding. Cardiolipin's a novel target, and when you target cardiolipin, it prevents assemblies of the proteins on that inner membrane, which causes that inner membrane to break down. We've further confirmed this through RNA sequencing and RT-qPCR. What we've also done is developed a novel method to turn the Galeria larvae clear, and we've monitored the clearance of the infection using optical microscopy, and this was again for Acinetobacter baumannii and Pseudomonas. Thank you.
Thanks, Kirsty. Very nice presentation. Again, if people can post their questions on the chat, that would be great. Kirsty, can I ask you a question also raised by Martin? Do you see much in the way of toxicity in human cells? Particularly, I was struck by the fact it was binding cardiolipin. So do you see any mitochondrial toxicity?
So we do see binding to mitochondria when we've done microscopy in eukaryotic cells, but they're not toxic. So the compounds IC50, average IC50 is 100. Well, they are toxic, but at much higher concentrations. So it's 135 micromolar for the dinuclear complex, which obviously when you compare that to the 0.5 to 5 micromolar that it's active on the bacteria, there's a large window there.
Yeah, okay, very interesting. And can I ask, presumably then limited to LPS bacteria, bacteria with LPS on the outer membrane? Is that...
Yeah, so we do see a higher activity in bacteria with LPS on the outer membrane. So we see lower activities with staph than we do for gram-negative bacteria. And then another reason being that it binds to teichoic acids and lipoteichoic acids in staph, which prevents a lot of it getting inside the cell. So that's the reason that we see a difference then in that sense as well.
I see. There's a question here from, moving up, sorry, from Jess. What are the next steps for Metallobio? How are you going to proceed to the clinic?
So currently we've just put in a grant for preclinical development, working with the MDC on preclinical imaging as well. So we're really going to determine the PK/PD and also do in vivo target indication efficacy. We're also going to look to do, so we haven't done any sputum testing or ex vivo testing in that sense. So we need to look to do something like that as well. And there are most immediate next steps.
Great. And sorry, one final question then. Sorry, you said it was luminescent, but it looks fluorescent. Did you mean fluorescent?
No, yeah, so they're luminescent.
Luminescent.
Yeah.
Interesting. Great. Well, look, if there aren't any other questions, thanks again, Kirsty, for a great talk, and good luck with the project. Wonderful. So the last of our 4 winners, Simone Lucanto from the National Biofilm Innovation Centre and University of Nottingham. Simone completed his BSc in biology and MSc in molecular biology at Roma Tre University, and then master's projects in the University of Nottingham. And I think He's going to talk to us about single chain antibodies able to recognize Pseudomonas quorum sensing molecules, I think. Simone.
Yes. So first, thank you very much for inviting me. I'm very delighted to have the opportunity to present my work today. So yes, Simone Locanto, I'm a second-year PhD student in microbiology at University of Nottingham within the National Biofilms Innovation Center. Next slide. Today, I will be presenting an antivirulent approach to tackle Pseudomonas aeruginosa infection by targeting the quorum sensing system. Next slide. So, I mean, we all know the importance of Pseudomonas aeruginosa and why it is essential to develop new strategy to fight this organism. Pseudomonas aeruginosa evolved a cell-to-cell communication system based on diffusible molecules called quorum sensing, which plays a key role in regulating the virulence of this pathogen. So it is very important to study this system, as disrupting quorum sensing would attenuate the virulence of Pseudomonas aeruginosa without imposing selective pressure, so differently from a conventional antibiotic. And for this reason, quorum sensing is a desirable antivirulent target. Next slide, please. So Pseudomonas aeruginosa possess three main quorum sensing systems, the LAS and RHL, that are driven by N-acetylmoserine lactones, whereas the PQS system uses alkylquinolones. We are focused on the alkylquinolones as they are very important for the pathogenesis of Pseudomonas aeruginosa. They're involved in the regulation of the biofilm formation and maintenance. Also, they regulate the production of virulence factors. And also, the single molecule possess additional properties. They are iron scavenger and immunomodulators. Also, we are very interested in the PQS system as we know it is active during infections. In a clinical study, we have been able to detect alkylquinolones in all the body fluids of CF patients, sputum, urine, saliva, and plasma, whereas the N-acyl or moserine lactones have been detected only in sputum and a very low concentration. So our approach is to quench these alkylquinolones using antibodies that can sequester these molecules and thus switch off the PQS system. Next slide, please. So here is a summary of our work. So we, in collaboration with the Scottish Biological Facility at the University of Aberdeen, we developed single-chain monoclonal single-chain antibodies, so monoclonal antibody fragment, against the two main alkylquinolones in Pseudomonas aeruginosa, PQS and HHQ. We have been able to successfully purify all these single-chain antibodies. Then we look into the sensitivity by competitive ELISA. These single-chain antibodies have shown a sensitivity in the low nanomolar range, which is in line with the level of alkylquinolones detected in body fluids. Most interestingly, then we looked into the impact of the single-chain antibodies against the PQS system using a Pseudomonas aeruginosa biosensor reporter. What we found is that when we had these antibodies, we were able to quench the quorum sensing response. However, we realized that we were not able to do it fully, as the single-chain antibodies are susceptible to Pseudomonas aeruginosa proteases. So what we are currently doing now, we are converting the single-chain antibodies into IgG, mouse IgG, as they should be more stable and they should show a higher affinity. So we are hoping that by using IgG, there will be a much more therapeutic impact. And this is where this project is currently heading. Next slide. Yes, in conclusion, I would like to thank my founders, the National Biofilm Innovation Center, the Cystic Fibrosis Trust, of course, my supervisor, and our collaborators at the University of Aberdeen. Thank you for your attention, and I'm happy to answer your questions.
Simone, thanks very much for a great talk and super quick. So there's plenty of time. No, it's great. Great. No, don't get me wrong. That's brilliant. Lots of time for questions. Can I ask why you went down the single chain antibody route to begin with, rather than kind of other types of nanobody or just the full length antibody to begin with? And I guess linked to that, do you see any benefits of having an FC region that will interact with host immune cells?
Yeah, absolutely. So basically, the idea of using first the single-chain antibodies is that they are easy and cost-effective to be produced. So basically, we currently produce these single-chain antibodies in E. coli, so they are very cheap and easy to purify. So the first screening was using these single antibodies to just select the best. Of course, having an all IgG that possess also the Fc part, it will be more effective as it would interact with the immune host, which is a thing that a single antibody cannot do.
Great, thank you, Simone. Some questions coming through then. First question from Leshan. Simone, how effective are these antibodies in terms of penetration to biofilm?
So great, thank you for this question, very important. Yes, this is a thing that we will look into. We have not done it yet, but we will definitely do, because actually it will be important to see the difference between the single antibody, the IgG, to penetrate the biofilm. We have not done it yet, but yes, we will definitely do.
It's on the to-do list. Great. A question from Catherine here. Do you know if the epitope is conserved across clinical samples?
So so basically, you mean if we can detect the same alkynolones in all the body fluids? So
the question is, I think, is the antibody effective at binding compounds across clinical isolate space? Yeah.
So we have not tested this antibody in clinical isolates. We are currently working using P1L laboratory strain, but we'll definitely involve also clinical isolates. Yes.
But the
important thing to know.
Sorry, carry on. Sorry. No, carry on.
The important thing that I would like to highlight is that, as I showed before, the Pseudomonas aeruginosa possess 3 main quorum sensing systems. But interestingly, in the clinical isolate, you see most of the clinical isolate are AQ producer, whereas most are lack the last system. So we believe that the alkylones have a more impact during the infection. And also, since we found in all the body fluids, they're more interesting.
Great. Simone, final question then. So from Sebastian, interesting targets to focus on. How do you think this could impact the regulation of virulence in pathogens?
So basically, it has been demonstrated that inhibitors of the BK system can downregulate quite massively the production of toxins like pyosanin, which is quite important during the infection in the lung. And also, interestingly, inhibiting the PK system sensitize the biofilm of Pseudomonas aeruginosa to antibiotics. So if you look at the inhibition of the quorum sensing, if you look in a synergistic therapeutic with antibiotics, that will be quite interesting, because if you can sensitize the biofilm to an antibiotic that is used for treatment, that would be fantastic. Yeah.
Great. So Simone, I said it was the last question. There's one more question from Deborah O'Neill. So since she's the boss, she gets to ask another question. I know it's early days, but is the focus on control or eradication? And how might you see this being administered in terms of formulation, duration, and frequency for benefit?
Yeah, so basically it is that with this antibody will be ideal to prevent. So basically, if we can inhibit the speaker system, we can prevent the formation of biofilm. And so basically, we will avoid the switch to chronic. So if you see in the literature, monoclonal antibody have been successfully nebulized, so delivered with nebulization. So which is something that we should look into in the future. Before doing that, we should also humanize these antibodies, because I would like to highlight that these are sheep-derived single antibodies. We are currently moving to mouse IgG, and then if they show up good therapeutic potential, we will need to humanize the IgG, and then we can look into also how to deliver these antibodies.
Terrific, Simone. Thank you. So I think it's a wrap now. So just thank you to all of the speakers for 4 great talks, and I'm going to hand back to Jess.
Thank you so much, Andres, and all of our speakers. It was really interesting, and I love the chat that was going on in the chat function as well, so thank you so much. We are now going to go into our breakout rooms. So we're going to have around about 40 minutes if we need it. If we don't, we can close them early. If we want longer and the discussion keeps going, then we'll keep them open a bit later. And what we really wanted to do through these breakout rooms was to get some open discussion on key topics. So one of them is around the CF AMR biorepository. So people can learn how to access samples, but also give us some information that is going to guide our future direction in the area. And then the second breakout session is around the how we can support antimicrobial development in CF. And in particular, we'll be thinking about the network and what we can do through the network to support you as a community. So we'll go into the breakout session soon. They're going to be really interactive. So if you can keep your webcam and your mic on, feel free to get involved in the chat, write in the chat function as well. Seema, I'm going to ask you to press the button and separate us. Hopefully we'll all separate quite well. If there's anybody that's left behind, Seema will shortly get you to the right breakout room. So if you can press the button, I'm going to click join now, which I assume everyone else has, and see you soon. So thank you so much.